going to be talking about some work I did as a postdoc at the University of Wyoming. I'm going to be talking specifically about contemporary hybridization between species of trout, specifically between rainbow trout and Yellowstone cutthroat. So, as some other speakers in this session have touched on, hybridization is common in nature, but we're still very much figuring out the details and mechanisms that underlie a lot of patterns that we see out in the world. The example I'll be talking about today is hybridization between native Yellowstone cutthroat trout and introduced rainbow trout. And our previous work shows that one really interesting feature of this system is that hybridization varies across locations within the same drainage where species come into contact, and also that we see this skew in ancestry of hybrid individuals towards the non-native parent herd rainbow trout, which is introduced in this region. And so we're seeing this variation, but we don't really know why. And so a lot of what we'd like to do now is to try and understand what drives hybridization dynamics. And so I won't talk about it much today, but my collaborators and I have a project looking at prezygotic mechanisms. There are two people in the field right now working very hard to look at overlap and spawning times. But what I'll be talking about more today is the potential for some sort of postzygotic mechanism that might shape which hybrids survive and might shape the genomes of those surviving hybrids in ways that are interesting for the evolution of these species and the nature of what we know about species boundaries. And so we have these kind of contradictory findings from a closely related subspecies from West Slope cutthroat trout that, on the one hand, individuals with more hybrid ancestry, more rainbow trout ancestry, produce fewer offspring in streams in Montana. Now, this isn't Yellowstone cutthroat trout, but it is a closely related subspecies, so it's a relevant example. But there's another recent study that suggests that there's convergence in which rainbow trout alleles rise to high frequency across multiple hybridization events in multiple locations, suggesting that there might, in some cases, be some rainbow trout alleles might be favored in the hybrids. And so this is kind of an interesting situation where we're trying to figure out how hybridization relates to fitness. So the questions I'm trying to address are how does hybridization shape locus-specific ancestry, and do we think this, to what extent do we think that this is potentially related to selection pressures? It's also important to note that loci could be behaving strangely for reasons that have nothing to do with selection, that in fact there are major differences in genome structure between these hybridizing species. Salmonid genomes are absolutely nuts for those of you who are not familiar with them. There's variation within a species in chromosome number, in location of the sex determinant region. It's just wild and crazy and lots of fun. And so the question is, how do two things that have different chromosome numbers even hybridize? And the answer seems to be that order of things is, things are not necessarily on the same chromosomes in different species, but they are in kind of the same order, enough that hybridization works. So for this particular project, and the one that preceded it, we sampled about 1,000 juvenile trout from 27 locations in the North Fork Shoshone River drainage in northwestern Wyoming near Yellowstone National Park. We then used a genotyping by sequencing method to produce about 2,150,000,000 base pair reads, which we aligned to the rainbow trout genome that is currently available. It's published as a preprint. And then from this alignment, we identified 25,000 SNPs, and these will be the basis of the analyses I'll be showing you today. So specifically, I'm using Bayesian genomic clines. And what this analysis does is that it looks at ancestry at a given locus relative to the overall ancestry of an individual. And we're trying to identify those loci that are behaving strangely in their genetic background. So loci that are introgressing into the other species background at unusual rates in some way. And so you can think of this as a situation where you could draw one of these lines for each locus throughout the genome. Our null expectation is this 1-1 line, the black one, where if an individual, for example, is about 40% rainbow trout, we would expect that there's a 40% chance that any locus would come from the rainbow trout, would have the rainbow trout allele, given nothing else going on. But we can have situations where either due to genome structure or selection, we might have a scenario where it's only, an individual is only perhaps 20% one species, but has a close to 100% chance of having the other species allele at a locus. So I said there are 25,000 SNPs. It would be cumbersome to try and draw one of these clines for each one of those. So I'll be showing the results a little differently. I'll be showing the parameters that dictate the shape of these curves. Specifically, I'll be showing alpha. Oh, and so we'd see with clines like the ones at the extremes here, we'd see either more rainbow trout than expected or more Yellowstone cutthroat than expected. And so when we plot genomic cline center alpha on the x-axis against genomic cline rate beta on the y-axis, we see a shotgun blast. But the peripheries of the shotgun blast have the potential to be interesting, and that's what I'm thinking about now. Note the differences in scale on the x and y axes. I'll be talking primarily about this x-axis because this y-axis doesn't really deviate substantially from 0 in ways that are likely to be interesting. And 0 is, again, that straight line of null expectations. So there's a skew toward rainbow trout ancestry, about a 5-to-4 ratio of extreme values toward rainbow trout relative to extreme values toward Yellowstone cutthroat, suggesting that there might be some sort of process skewing these individuals, these hybrid individuals, toward rainbow trout ancestry at some loci. As of now, we don't know why that is, but we can also look at this in a more explicit way across the genome and look at the actual location of these loci that are behaving unusually. So the loci that are unusually low are unusually high. The ones that are unusually high are skewed toward rainbow trout. The ones that are unusually low are skewed toward Yellowstone cutthroat. And so what you can see here is that it's not a large scale pattern. It's not that there's like half of a chromosome that's all skewed one way. In general, the genomes are introgressing they're mixing fairly well, and there don't seem to be these giant chunks that are purely ancestry from one species. But if we summarize that previous figure in terms of extreme values and look at number of extreme outliers per base pair, we can see that different chromosomes have different numbers of these outliers. And in particular, I think something that has the potential to be interesting is this chromosome 20. Chromosome 20 could be cool for a couple of reasons. One is that the rainbow trout preprint suggests that for the rainbow trout genome, there's a giant inversion in chromosome 20. So part of the chromosome flipped upside down, and it might contain potentially, they're not sure about the phenotypes related to that inversion, but there's a lot going on with sex-dependent loci being involved in inversions in this genome. We know that from previous work. But also chromosome 20 is one of the ones that we think is not the same structure in rainbow trout and in Yellowstone cutthroat. So it's possible that some genomic rearrangement is inhibiting or skewing recombination in this area. So this is work in progress. There's still a lot to do on this project, and I'm excited to push this further. But what we know is that the genomes are introgressing relatively freely, consistent with the fact that out in these systems we see lots of hybrids being produced, and suggesting that there aren't really that many hard limits. The overall ancestry is skewed toward rainbow trout, and we think that this genome rearrangement story might have some interesting implications for hybridization outcomes. And this could be either through chunks of ancestry in genome rearrangements could be preserved, like adaptive complexes could be conserved, or we could have a scenario where deleterious things are not being broken up by recombination, and so we could have some selection out of the population of individuals with those particular sets of alleles. This is what I was referencing previously, is that a linkage mapping project suggests that in hybrids, at least hybrid males in this instance of their study, that the linkage group corresponding to chromosome 20 also has loci that turn up on chromosome 28 in rainbow trout. So there is kind of this difference in genome structure. So I think that actually both these stories I started telling you at the beginning could be true. So I think that this story that admixed individuals are producing fewer offspring, that could be true, but it could be that the individuals that are making it through this reshuffling of genomes through hybridization as this variation generating machine could be selected in ways that skew toward rainbow trout alleles at some loci, potentially because it's favored for some reason. And so I think that that's kind of an interesting point that I'm going to pursue further. And so we'll be looking at more specifically at where those extreme values are, where those unusually introgressing loci are in the genome. This is a beautiful figure from that rainbow trout preprint showing differences in, I believe, recombination rate across the genome on the periphery, and then the connections of which regions of the genome are homologous. So we'll be doing some sliding window analysis to look at the genome in a more fine-scale way. And so I'm also going to attempt to identify sex-linked loci, because I think that sex-linked sex bias processes and hybridization could be particularly important for this system, especially given what we know about selection operating differently on sexes in rainbow trout. So stay tuned for more work on this topic. I'd like to thank the many funding sources. This work is heavily supported by the Wyoming Game and Fish Department and would not be possible without their ongoing collaboration. I'd also like to thank my collaborators at the University of Wyoming and the University for computing and logistical support. Also, I'm recruiting graduate students for my new lab at the University of Guelph. In particular, I'm looking for a student to work on the effects of anthropogenic disturbance on fish genetic diversity and hybridization rates. So talk to me if you're interested, or email me. Okay, thank you.
So you might have said this, but do the hybrids have differences in chromosome number? Is there variation? Or?
That's such a good question. So the question is whether hybrids have variation in chromosome number, and the answer is that we just don't know. So we might be able to get at this. So there's some suggestion from Osberg's work that hybrids match rainbow trout pretty well, but there's also not a Yellowstone template linked to that. And so that's a very weird thing. So we might be able to do something useful with recombination in hybrids, potentially. But it's, at this point, we're not sure.
Do your alpha outliers relate to any sort of climatic variables?
Sorry?
Do your alpha outliers relate to any sort of climatic variables? Like, so the allele frequency at SNPs which were identified as alpha outliers, do they relate to any sort of climatic variables?
Oh, whether they relate to climatic variables, that's a good question. And actually, I haven't looked at that. And I have the data to potentially look at it, because these streams do vary in temperature. And we see that overall hybridization dynamics in each stream are related to temperature. But as of now, I don't know. But that would be a really interesting thing to look at. Thank you.